Good afternoon. Good afternoon, everybody. Welcome to our seminar this afternoon here in Toronto at the MHRC at York University. It's a pleasure for me this afternoon to welcome Dr. Glen Tibbits from Simon Fraser University. Let me tell you a little bit about Dr. Tibbits' background. He's originally from Montreal. He went to McGill, got his bachelor's degree, Bachelor of Education degree at Montreal. McGill, and then went to UCLA, where he did his master's and PhD degree at UCLA. He did a postdoc in Japan for 1 year, then came back from that and worked as a research assistant professor at UCLA for a period of time, and then got a job at the University of Washington in Seattle. Spent about 15 years in the United States. But he was just telling me that he was longing to come back to Canada, and he got a job at Simon Fraser University, where he's been ever since. And it seems like about 35 years he's been at Simon Fraser now, where his position is a full professor and Tier 1 Canada Research Chair in molecular cardiac physiology. And to be honest, I've been reading Dr. Tibbits' papers since I was a grad student, learning about cardiac calcium and contractility and the effects of exercise. So it's really a pleasure for me to have him here, and I'm really looking forward to his talk, which is investigating inherited cardiomyopathies and arrhythmias using cardiomyocytes. So Glen, I'd like you to share your slides with us, please, if you would, and unmute yourself, and we're looking forward to your talk this afternoon.
Thanks very much, David. Can you see my slides all right?
Yep, looks good. Very nice.
And this is, yeah, thank you. I appreciate the invitation. The flight was a disaster, I can tell you. The waiting lines at the airport, and I don't know, that crappy hotel you put me in last night.
We'll have to do better. We'll have to do better.
I mean, I thought I'd be staying at the Royal York, but I don't know what it was. Anyways, it's a pleasure to talk to people at York. I've known many of you for a long time. And of course, I know Peter. I've known Peter for a long time when he was at U of T, and now at York. So as David mentioned, I'm going to be presenting our work using induced pluripotent stem cell derived cardiomyocytes to study inherited cardiomyopathies and inherited arrhythmias. So David told me that there's quite a broad background of people, so I'm going to give a little bit of a background. David, I don't know what the format is. Do people ask questions during the presentation, or are they all directed to the speaker after the presentation?
Well, usually what we do is wait till the end, and people who have questions go through the Q&A option there, and I'll actually read the questions to you so everybody can hear, and we'll do it that way at the end.
Sounds good. Okay. So, as I said, we study inherited arrhythmias and cardiomyopathies, and typically we're looking at point mutations in genes that, when they're harbored in patients, can cause, for the cardiac arrhythmias, typically they're causing channelopathies, cardiac ion channel dysfunction, and some of the channelopathies that we study, long QT syndrome, catecholaminergic polymorphic ventricular tachycardia, and atrial fibrillation. And because of the channelopathies that are associated with these and other diseases, we can get very aberrant ECGs. This ECG is from a patient with CPVT, which is exercise induced. And as they go into different levels of exercise, they start developing this very anomalous ECG that's biphasic, ventriculars, et cetera. It's kind of a disaster. And of course, this can result in ventricular fibrillation and sudden cardiac arrest. The channelopathies is something that's also very close to our heart. And typically these are mutations in cardiac contractile proteins. More specifically, we focus on the troponin genes. But interestingly, patients that develop these can develop hypertrophic cardiomyopathies or dilated cardiomyopathies or other types of cardiomyopathies with mutations in the same gene. Not necessarily the same mutation, but within the same gene. So there's 20 or so contractile proteins that are encoded for that give rise to the vast majority of these cardiomyopathies. But the interesting part of it is that patients with hypertrophic cardiomyopathy or dilated cardiomyopathy, they normally die from arrhythmias, from sudden cardiac arrest, which can be obviously very serious. So I've been working, our lab has been working on using native cardiomyocytes for many years. We've used native cardiomyocytes from rabbits, mice, rats, etc., even humans, which is a bit challenging, but we're able to get some atrial appendage tissue when they were doing open heart surgery on children, but it was a challenge. But one of the problems, as most people know, is when you're modeling inherited arrhythmias and you're using the murines, mice or rats, mice in particular have heart rates up to 700 beats per minute. They have a different expression profile of ion channels to accommodate those high rates. It makes it very difficult to really model human arrhythmias with the degree of veracity that one might like. And of course, humans represent a different type of challenge: access to tissue, controlling the environment of the cells. Most of the patients from whom you get cells have been on drugs, a variety of different pharmacological agents, etc. So we were pretty excited when the whole concept of human-induced pluripotent stem cells came into view with the really incredible work of Shinya Yamanaka. And what he did was he was able to take fibroblasts from patients and he was able to infect them with a retrovirus harboring what we now call the Yamanaka factors. These are transcriptional factors associated with pluripotency. And that would have caused reprogramming of these fibroblasts over the span of about 5 weeks, in which they became, well, as some people say, back to the future. They have now developed the ability to become anything they want, as people always tell their kids when they're growing up. With hard work, you can become whatever. Some of these cells might want to become cardiomyocytes, but this process of reprogramming that Yamanaka developed has allowed people to use these iPSC-derived cells for heart cells, as I'll talk about, neurons. My colleagues work extensively on pancreatic beta cells to treat type 1 and type 2 diabetes, and macular degeneration, etc., retinal cells. So this really incredible work was done by Shinya Yamanaka, kind of became my hero. He published this in 2006 in the mouse, 2007 in humans. And I really, when I read these papers, I thought, this is what I really want to do, because we had so many issues with native cardiomyocytes. But I really didn't feel like, we had any right to be working in the field of stem cells. It's not something I knew anything about. But anyway, in 2011, my CRC got renewed and I said, let's go for it. I had some sort of flexibility in my funding and my status. So we started in 2011. And then the amazing part was in 2012, Yamanaka won the Nobel Prize with some other stem cell biologists. But this work really was transformative in that early stages, and recognized so that he won the Nobel Prize within 5 years of this kind of impressive discovery. So I've been very fortunate to get into this field early, and we started by developing a lot of the critical assays, as I mentioned, 2011, 2012 at Simon Fraser University. But then people ask me to come to Children's Hospital, where they're really invested a lot of money and a lot of resources into developing what we call the Cellular and Regenerative Medicine Center. And I'm just going to show you the pipeline that we've developed that we use. So we can take PBMCs, monocytes from patients. We can reprogram them in ways slightly different from what Yamanaka did. We can expand this population and make our own iPSCs, or we can use iPSCs that we've curated. Genome editing, so this gives us the opportunity to introduce homozygous or heterozygous mutations, and we have these modified iPSCs. And of course, genome editing has just been completely transformed by the CRISPR-Cas9 work of Jennifer Doudna and Carpentier, and they won the Nobel Prize this past few months ago. Incredible work and just transformative as well. So this is how we do. We take our iPSCs, we infect them with a plasmid that contains Cas9, which cuts the DNA, has a GFP reporter on it to tell us which cells actually got transfected, and includes guide RNAs that direct the Cas9 to the appropriate place in the genome under their own promoter. And then we can look at the cells that were infected, and then we can screen for those which have undergone homology-directed repair. So the DNA gets cut, and then it invokes its own repair mechanisms. And we have to look for the needle in the haystack, because homology-directed repair occurs relatively infrequently. Maybe 2 or 3% of the cells repair themselves in a way that will generate these end products. And so we have ways of doing this. We've switched recently to a ddPCR assay that Bruce Conklin developed at the Gladstone Institute. And it's pretty cool. So we can isolate the heterozygous mutants or the homozygous mutants, as well as the wild type that undergone exactly the same process, but end up as wild type. So this would be our isogenic control. And we would always be comparing our mutant with our isogenic control that's undergone the same process. And then once we have these colonies, we can differentiate them after extensive quality control, which I won't go into cardiomyocytes. So I hope that makes some sense. It's taken a while to evolve in a way that allows us to get some fairly high quality cells. But in addition to that, we can also take curated lines, which we have. We have several of them from various institutes, like the one in Wisconsin, the Allen Institute in Seattle, which is just an amazing place that was developed from Paul Allen, who used his money from Microsoft to not only buy the Seattle Seahawks, but also to develop these Allen Institutes, of which there are 3, I believe. And they've made lines with a variety of different reporters in that we use. So, and we also generate our own reporters. So we CRISPR in GFPs into proteins to follow them. And then after quality control, we can biobank these right here. We then go through this process of differentiating the cardiomyocytes, then enriching them to increase the percentage of cells that are actually cardiomyocytes, and we can get this number up to 90% or so. And there's a variety of ways you can do this, which I won't really talk about unless someone's interested. But this process of cardiac differentiation has evolved in our field, so it's become fairly sophisticated. And the whole process here is to try and recapitulate what happens in utero. And that's kind of shown here. We have pluripotent stem cells from which the heart is made, and it goes through a variety of steps, and the transcription factors or the genes that are expressed typically in each of these stages is shown here in black. And so we try to guide it through this process by, first of all, activating the Wnt signaling pathway by disinhibiting it using this CHIRE small molecule. Inhibits the GSK3B, activates the Wnt signaling pathway. And then we get it into this mesoderm stage, and then we set about by inhibiting the Wnt signaling pathway a few days later, and we get into this cardiac progenitor stage, and then ultimately we end up at these cardiomyocytes, which are expressing most of the genes. As you can see, we have myosin heavy chain, troponin T, troponin I, etc. And these cells are beating, so that they look like cardiac cells in many respects. However, they lack the full maturity of an adult cardiomyocyte, and that immaturity is reflected in the absence of T tubules and some other things that I'll address in a moment. But through this manipulation of the cell media, we can get to this immature state, and with enrichment, we can get ventricular cardiomyocytes to represent about 90% of the population. And just to show you, this is a 35 millimeter plate that my PhD student, Sanam Shafatalab, developed a lot of these assays at SFU many years ago. And so these actually started as PBMCs from her blood sample. So a phlebotomist took her blood, she isolated the PBMCs from her own blood, she reprogrammed the PBMCs into iPSCs using a variant of the Yamanaka process. And then she started differentiating her iPSCs into cardiac myocytes, following this pathway that I just talked about. And if you look at some of the markers during this process, NANOG and OCT34 are two pluripotent markers, and they're expressed at very high levels just before she started the differentiation protocols. And almost immediately within a day, these things fall to just a fraction of what they were previously. So in other words, the cells are losing their pluripotency as soon as she begins to differentiate them into cardiac myocytes. And conversely, troponin T and troponin I start taking off and increasing very dramatically shortly after the process of differentiation. So when she first did this, 2013 or 14, whatever, the thing that she noticed after 12 days is that the whole Petri dish was beating. Now, you've probably seen this before, but there's something here that's different from what you might have seen before. And that is this Petri dish, which is 35 millimeters, is actually showing a very highly orchestrated beating. And you'll see later on, there's a very clear conduction velocity across the plate, reflecting the interaction of the cells in here. So this might contain hundreds of thousands of cells that we've made into cardiac myocytes. So Sanam did an amazing job in developing this. She's now a postdoc in the Bill Poo group at Harvard using iPSCs to study CPVT, and doing a great job there as well. So as I mentioned before, the iPSC-derived cardiomyocytes that we develop in culture are immature in several respects. So this is a mature cardiac myocyte here, showing the typical sarcomeric pattern with a spacing of around 2 microns, and T tubules, which you can't see in this type of image. And these are missing in the immature cell, in the iPSC-derived cardiomyocyte. The T tubule is missing, the very highly orchestrated array of L-type calcium channels, the more developed structure of the SR in conjunction with the T tubules. These are somewhat missing. However, they do express SR, they do sequester large amounts of calcium, but the interface between the L-type channel and the SR is not fully developed. How can we go from the immature cell to the mature cell? Well, it turns out we've learned a lot about this process. And one of the processes, or a class of process, is to use chemicals. T3 from the thyroid gland is known to cause maturity. Chuck Murray in Seattle has shown this. Free fatty acids, a variety of different substrates can enhance the maturation level. In fact, there's a hypothesis that's been developed recently in which a variety of factors, which by inhibiting the mTOR pathway, induce the cardiac maturation and the quiescence that we like to see in a cell that we hope will recapitulate an adult or a mature cardiac myocyte. But in addition to that, we know that electrical stimulation, depolarizing the cell at high frequencies for a period of weeks, can also induce maturation, nanotopography of the extracellular matrix, and mechanical stretch, mechanical deformation, a variety of things that can be imposed on the cell depending on the environment in which they're placed. So this is a very hot topic, obviously for big pharma and for people who want to understand adult cells. This is really crucial. And so there's lots of work that's being done. A lot of it's in Toronto, by the way. As Peter knows, Peter's interacted and published with a number of people who are focused on this aspect of the maturation protocol. So we have a protocol in which we have cardiomyocyte differentiation. Then we go through a separate protocol of not just differentiating, but maturing the cells by changing the substrates and the environment in which the cells are growing. And once we have this, we next go into a tissue engineering phase. Again, this is something that Peter has worked with really prominent people in U of T who are doing tissue engineering. I'll tell you a little bit about what we're doing in this regard. And then we do extensive phenotyping of these cells, going all the way from the genome to the transcriptome, the proteome, and a whole variety of phenotypic studies from metabolic assays such as seahorse to structural studies using electron microscopy, optical mapping, patch clamping, you name it. This is just some of the phenotyping that we use. So one of the most important phenotyping protocols we use is optical mapping. And in the optical mapping, we load the cells with typically two dyes. One that is a potentiometric dye, responds to changes in voltage, and the other is a calcium sensitive dye. And this was developed largely by Dr. Glen in my lab, who got his PhD a number of years ago and really is an imaging specialist. So he built our optical mapping rigs. He's written the software, which is really sophisticated, and it allows us to look and unmask what these mutations are doing in terms of cardiac excitation or even their contractile properties. So as I mentioned before, one of the unique things about these beating cardiomyocytes that I showed you in our dish was the fact that they have very high conduction velocities, which has a vector to it. They're moving in a specific direction. If they are stimulated with like a point stimulation rate here, they propagate very easily. And if you go on the internet and you look at beating cardiomyocytes derived from hiPSCs, typically what you see are little patches of beating cells. So these are clearly developed, intercalated discs with gap junctions. They express connexin 43, but more importantly, they're functional based on the conduction velocities that we're showing. And again, this is the rig that Eric developed, and these are the dyes. So the black is showing the change in voltage with a potentiometric dye. In this case, we're using RH237, and the red is showing the calcium transient shown by rod 2, and the camera is capturing this, and we can either stimulate the cells at a given frequency, or we can look at their spontaneous beating rate. And this is the really sophisticated part of Eric's optical mapping rig, in that we can establish multiple regions of interest across this plate. And these are shown in a cartoon fashion as a blue region of interest, green, orange, and a black. And we can look at the calcium and the voltage in each one of these regions of interest independently. And we can look at the relationship between these two in terms of when these events are occurring. So we can look at conduction blocks, we can look at a variety of different signals. And the amount of information that's coming off of this 35 millimeter Petri dish is in the order of 1 gigabyte per second. Huge amount of information. And Eric had to work out a lot of the details of how the computer could actually cope with that kind of information. So one of our first applications was this interesting study in which the medical examiner of Manitoba asked us to look at tissue samples from 191 deceased infants. And these infants were all autopsy negative, meaning that they had clearly, after death, they'd undergone extensive autopsy, toxicology screens, X-ray analysis. Everything was negative. There was no clear indication as to why these infants died. And most of them died within 2 years of age. The reason we were involved in this is that one of my PhD students, Laura Dewar, was actually a coroner in BC for 20 years. And so she had rapport with the medical examiners across Canada. And so obviously in Manitoba, they were really upset by the fact that they had so many samples from from infants from whom they had no idea actually how they died. So they sent us the samples, and we had set up a panel of genes that we thought might be indicative of genes or mutations in genes which could result in sudden cardiac arrest as a potential cause for their death. And we found quite a few mutations that have already been associated with sudden cardiac death, ones that would be indicative of long QT and perhaps CPVT, other ones. We didn't pursue those because we did find something that was really interesting, and that was that 10 of the 191 infants were harboring a mutation in troponin I1, R37C, a heterozygous mutation, or variant, I should say. Excuse me. But the interesting part is that this form of TNI is actually only expressed in the infant's heart. It's referred to as the slow skeletal TNI. And after about 2 or 3 years of age, we express almost exclusively the cardiac form, and the gene name for that is TNNI3. So it looks like these infants had a mutation that affected them because they were expressing it in the heart. And perhaps this was a very serious mutation, although it was unknown at the time whether this variant was pathological or not. What we did do is we looked at it in terms of a phylogenetic analysis, and we could see that the arginine at position 37 had been conserved over 400 million years of evolution. It was still in the zebrafish. The next thing we did was we used molecular dynamics and molecular dynamic simulations, and we did this in conjunction with Peter Tillman and a postdoc in my lab. Charlie Stevens did this by modeling the troponin I, troponin T, troponin C complex with and without the mutation of interest. So in molecular dynamic simulations, you start with a crystal structure, a known structure, and then you make these mutations, and it uses a program called GROMACS, and then it recalculates the thermodynamic environment for every one of the interactions of this with every possible. So the calculations are unbelievable, and it goes on to Compute Canada, or in Western Canada it was the West Grid, and it may go on there and do the calculations for 2 or 3 weeks. But what we found was that when the arginine at position 37 was mutated or was a variant of cysteine, we had a huge difference between the interrelationship between troponin I and troponin T here. So this is the wild type. And what we modeled was there were a lot of H bonds that existed between the troponin T and the troponin I. And that was largely because of the arginine right here. But when that was mutated to cysteine right here, it lost this interaction. So again, in addition to the phylogenetic assay, we thought this was a serious mutation that is likely pathogenic, but we wanted to go ahead and convince ourselves that this might have been involved in the death of these infants. So the next thing we did was we created or reconstituted cardiac thin filaments And this was done by another PhD student, Allison Lee, in my lab. And what she did was she had the human genes for troponin C, troponin I, troponin T, and tropomyosin. And she extracted rabbit skeletal actin, which is a 98% degree of homology. And she was able to reconstruct the thin filament by putting those recombinant proteins together in the appropriate stoichiometry. And we could check this, the function of this, in a variety of ways using fluorophores. Anyway, what we found was that she could make both the adult and the neonate while a recombinant thin filament. The neonate, which reflects the infants that we were looking at, have two major differences. They have differences in the TNI paralog that I mentioned, the switch from the slow skeletal to the cardiac, or as they mature, but also there's a different splice variant of the troponin T. So she was able to make both an adult form and a neonate form with those two changes I just mentioned, and then with or without the mutation of interest. And what she found was that the off rate constant, the rate the calcium comes off of this complex, was decreased in the neonate, but was increased in the mutant neonate that was the same mutation harbored by those infants. So the next thing that we did, again, we had pretty convincing evidence that this was not a benign variant in the troponin I. So the next thing we did is we took hiPSCs and we CRISPR'd in the troponin I mutation of interest. And then in the Petri dish, we could stimulate them at different frequencies. So again, the red here is showing the voltage, the blue is showing the calcium. We can stimulate these to a higher frequency. We can take them up to 120 beats per minute. And these tend to get entrained completely without any indication of abnormality at the higher frequencies. We do see some important aspects, though. The action potential and the calcium transient get shorter as the frequency gets higher, which is an endogenous aspect of cardiac function, which is very important as the human heart goes, from a resting heart rate to a higher heart rate. So when we looked at the isogenic, these are the isogenic controls. When we looked at the isogenic equivalent, but with the R37C CRISPR'd into these cells, they behaved reasonably well at 55 beats per minute. But at 75 beats per minute, we saw this clear sign of alternans, which is clearly an arrhythmogenic situation, which can degenerate into ventricular fibrillation. And as we took it to a higher frequency, here it's only 80 beats per minute. The cells could not tolerate going to a higher frequency than this. We see a much more severe alternans. The green indicates where we actually stimulated, but almost nothing happened. So clearly, this was a very strong indication that this mutation was likely the cause of sudden cardiac arrest and likely the cause of death in these infants in the crib because of the mutation that they were harboring. Now, the action potential, as I mentioned, gets shorter as the beating frequency increases, and this graph shows a beating frequency moving in this direction. This is a higher beating rate than this. This is the interval between beats. And this we call action potential restitution, a normal process in the heart, which allows the action potential to get shorter, allowing higher heart rates. The mutant, however, showed almost no restitution and the inability to go to heart rates above this right here, which I showed on the previous slide. So one other thing, we're doing a variety of things, but something that we were also interested in, one of our colleagues who moved from Toronto to British Columbia, Zach Blacksman, was interested in atrial fibrillation, as are we, as is Peter, as am I. Peter and I have very personal reasons for being interested in atrial fibrillation, but we wanted to be able to make atrial cells from our iPSCs. And Gordon Keller, who's also based in Toronto, a really stellar cell biologist, had developed a procedure for embryonic stem cells. We chose a slightly different process based on the same principle, but a simpler process, a little more reproducibly done. And it's simply adding retinoic acid during the differentiation process over a 3-day period at a fixed dose of 0.75 micromolar. And so the reason that we want to do this is that we want to get iPSCs from patients that have what we call lone AF. And lone AF is a type of AF that's developed in the absence of comorbidities. So these patients don't have a lot of the other factors that often induce the AF, such as heart failure or fibrosis, or a variety of things that could cause atrial fibrillation secondary to the actual disease. And the other thing that we want to do is to take GWAS studies of looking at the coincidence of genetic variants with the incidence of atrial fibrillation. So Zach had patients with lone AF, and we wanted to be informed by his patient base to use iPSC and then CRISPR in mutations of interest. So we have the lone AFs, which we haven't really started on, but we have been working now on the GWAS identified variants. One of the main players in this field is Patrick Eleanor at Harvard, and he's developed a variety of different GWAS identified atrial fibrillation variants, of which has informed some of our work. So when we put the cells through our protocol, what we found in our atrial specific population, which are shown in red compared to our ventricular or the normal population, we could see an increase in KV 1.3, which is a nodal atrial specific expression, the inward rectifier 3.1, KV 1.5, which is an atrial specific potassium channel, and SK3, which is controversial. This is not a voltage gated potassium channel, but a calcium activated potassium channel. But we found it was upregulated, but wasn't significant. We also developed a code set of 250 transcripts, which we could study using a technique called nanostring. And so we could look at the differential transcriptome between the cells that were pushed down an atrial lineage versus a ventricular lineage. So this is a volcano plot. If you're not familiar with them, this is showing the magnitude of the difference in this direction and the significance of the difference. And shown in red here are upregulation, significantly upregulated genes. And the red dots each represent a transcript that is atrial specific, such as KV 1.5 right here, and a variety of other atrial specific genes which are upregulated, consistent with what we had hoped to see in our atrial population. And then we began to look at the functional properties using optical mapping techniques. And if we look at the atrial action potential shown in red here versus the ventricular action potential, we can see a clear shortening, which is reminiscent of a native atrial cardiomyocyte, and a shortening of the calcium transient shown right here. This is a patch clamp that was done by our colleague, Vasilios Bezaridis at Harvard, and they show clear differences between the atrial down here and the ventricular, but they also show some of the immature aspects, and that is the resting membrane potential is around minus 58 millivolts, but they're also consistent between the atrial and the ventricular cells. Also, the upstroke of the action potential is lower than what we'd expect to see in a mature cell for a variety of reasons, which we could talk about if you're interested. But one of the interesting things was we wanted this to be a useful tool to look at atrial specific drugs, in particular Vernakalant, which was developed at UBC as a potential drug for treating atrial fibrillation to cardiovert patients who are in AF to go back to sinus rhythm. And Vernakalant is as our most antiarrhythmic drugs, a fairly dirty drug. One of the targets is Kv 1.5, which is the atrial specific potassium channel, but it has other targets as well. So what we're able to show is that our atrial cells had a clear dose dependence. So the lighter color is 0 Vernakalant. The higher concentration is up 30 micromolar, a clear prolongation of the action potential, which is due in part to the inhibition of Kv 1.5. And this was not seen in our ventricular cells. So in fact, we saw, if anything, a shortening of the action potential, but only at very high doses. In essence, we saw no difference in ventricular cells plus or minus this drug. This is thought to be antiarrhythmic or might help the situation of AF because broadening of the action potential might increase the refractory period and decrease the propensity to go into high frequency or fibrillation-like environment. So coming back to our Cellular and Regenerative Medicine Center that we're developing here at Children's Hospital, it's developed into a pipeline which is now fully functional. And we've tried to develop as a regional resource so that we could not only generate PBMCs and hiPSCs from patients, we could biobank them. We're also set up to look at hiPSCs from our colleagues at Harvard who are looking at patients that have CPVT but are resistant to an important drug in their treatment called flecainide. And so they're isolating PBMCs and they are sending those to us so that we can differentiate them into cardiomyocytes. Now, we've been really, really fortunate, and that is that the Children's Foundation has invested a fair amount of money, but very recently, we got money from this group, Mining for Miracles, and as you probably know, BC has a lot of very strong mining industry, and they have made a contribution of 3.4 million to our center here. And this has gone to several things. We haven't spent all the money yet, but we've increased our phenotyping. Actually, Peter has been involved in part of this. Aurora, as in Aurora, a suburb of Toronto, makes an atomic force microscope to measure the force of contraction of myofibrils, and we have purchased theirs. It still hasn't arrived because of the COVID. They had some issues with the construction. We've also purchased this Aspect 3D bioprinter. This is a local spin-off company, and what it allows us to do is to mix hiPSC-derived cardiomyocytes with fibroblasts, endothelial cells, with various extracellular matrix media. They're called bioinks in this environment. So we can engineer a fairly reproducible tissue chip that we're doing in conjunction with our colleagues at Harvard, in particular, Kit Parker, who's done some incredible stuff in tissue engineering. We are adapting our techniques or our equipment to invoke some of his very sophisticated techniques. And lastly, we're just in the process of roboticizing our whole process of differentiating cardiomyocytes so that this Hamilton Vantage will be part of a bigger system, which will include incubators, imaging systems, so that it can go through this entire process of differentiating cardiomyocytes. And the reason for this, and this is something that Peter and I have talked about, is that we want to look at mutations in multiple genomic lines. And this is one of the reasons why we want to roboticize this process so that we can in fact run 5 or 6 different genomes representing 5 or 6 different individuals with the same mutation to look at sort of the in vitro equivalent of penetrance of a genetic variant. What is the degree of pathogenicity, if you will? So just to summarize, these are the caveats. And it's interesting because when I started in 2011, the caveats took up 3 slides. There were so many issues that needed to be addressed. And the amazing thing about this field is it's so intense. Things are changing on a daily basis. However, when you're isolating iPSCs from patients, quality control is really critical and often ignored. And there's many aspects to this, including genome integrity, pluripotency, contamination, etc. Even after this process of quality control, the differentiation can result in heterogeneity. I mean, I shouldn't say can, does. But we know this from single cell RNA-seq that even in a native heart tissue, we have a huge variety of cell types, even within the ventricular wall, even within the endocardium per se. So this is not unique to the in vitro work, but it's something we have to think about. And maturation, although it's a focus of many labs, very intense work, it's something you have to be considering all the time to interpret your data. One of the beauties of this whole technique is the amenity of tissue engineering protocols. And the tissue engineering field is just simply taking off at an incredible rate, where people, as you know, are making organoids, they're vascularizing their tissue chips. And so this is something that we are part of. We're not a leader in the field by any stretch. It's a very complex field, but it's really exciting and it's really fun to be part of. And this is something that people need to think of. And that is when you disease model like we've done, like others, you're often using a single genome. And this is not unique to iPSCs. If people are using CHO cells or HEK cells, they're typically dealing with only one genome and they don't think about it. I didn't really think about it much until we started doing these iPSCs, and we get them from a specific individual, man, woman, Asian, Caucasian, what have you. They have a very specific genomic background, which we have to consider. But of course, the techniques offer tremendous advantages, I think. And we can give us insights into cardiology. In regenerative medicine, our colleagues in Seattle, like Chuck Murray, has FDA approval to be injecting embryonic stem cells into patients post-infarct. This is not something that I want to do. This is a really challenging field, but people like Chuck Murray are doing it and they're showing some promise. Drug development is something we do and something I alluded to. And what we focus on more than anything else is trying to understand a patient's pathogenicity of the mutation and how them with their particular genome may be at risk. So a type of risk stratification, which I'll come back to. But I can't emphasize enough how exciting this field is in that we have so many disciplines impinging on the kinds of experiments that not only we need to do, but we can do. And it's obviously a powerful technique for a disease in a dish. We can hopefully eventually risk stratify patients and determine the best therapeutic intervention and drugs which are contraindicated against that genome, etc. So I'm going to finish here for now and just thank all the people that did all of this work, our colleagues from all over the place who have just taught us so much. And it's been such a pleasure to interact with them, that it's a constant learning process and a really exciting learning process. So I thank you for the invitation, and it was a pleasure to fly to Toronto in the beginning of winter.
Glenn, thank you so much for that. That's wonderful. I have a new appreciation for tissue engineering. I think we're going to take some questions, and if you don't mind, please hit the Q&A button and ask your questions of Glenn. If you don't mind, we'll do it that way. Glenn, you must have the top cardiac muscle tissue engineering lab in the country. I mean, that is clearly, is there anything more advanced in the country?
Yeah, it's in Toronto. So Peter, so Peter interacts with a variety of people who really are tissue engineers. They're bioengineers and they've had a long history. We are neophytes at this field. So we rely heavily on interacting with people who are actually educated as engineers and tissue engineers. So we're making use of their discoveries. We're not leading the field. We're simply trying to use as a tool to get a better physiological environment or a tissue chip, which might be more reflective of the physiological state. So we're not the leader in Canada by any stretch, but we're
Getting there. You're getting
listening
there.
to them.
Let's take Peter Bax's questions here. Okay, so what method are you using to promote maturation? Because one of the main challenges in these cells is maturation. How are you promoting maturation?
We're using a couple of techniques. One is called the Albumax, which is a variety of free fatty acids clustered with an albumin molecule and some other aspects, some other chemicals in there. We're also using mTOR inhibition, so taurin-1, rapamycin, those kinds of things which inhibit mTOR. They also produce it. But we're looking at maturation from so many different perspectives. Like we're collaborating with Ed Moore on the EM, the development of the T-tubules. We're looking at mitochondrial proliferation. We're looking at, increased beta oxidation. There's so many facets to maturation that we may require several different maturation protocols to get where we want to go.
Another question is, the Bax group finds it's challenging to get tissues with ordered intercalated discs as well as sarcomeres. Do you see intercalated discs in your imaging assays?
Yes, we do. We see them, and not only do we see them, but we see the functionality of them. So in terms of the electron microscopy, is that what Peter's asking?
That's not what he mentioned, but whatever way you see them, I guess it would be with EM.
Yeah, so we do see intercalated discs, but we do not see them in a way yet that you would expect to see in a native cardiomyocyte, where they're very clear and you have clear differentiations, a box-like, rod-like structure of a cardiomyocyte. We're not there yet, but we're heavily engaged in trying to get to that stage.
Right, right. Shivam is asking, can your techniques be applied to acquired arrhythmias such as those from IR injury? Why do you think you see divergent effects in atrial cardiomyocytes versus ventricular myocytes?
With acquired arrhythmias, is that what the question is?
Can your techniques be applied to acquired arrhythmias?
Yes, so what we would do, so we think that acquired arrhythmias, the classic example is long QT syndrome using, antihistamines over the counter, giving long QT, which is kind of the reason why the FDA now requires people to look at drugs and their ability to inhibit hERG. But we can use drugs to see what their effects are on the exponential duration, on the optical mapping protocol, and see if they're likely to induce an arrhythmogenic response. So we could look at acquired, we can study them over a period of weeks and see whether there's an acute effect or something that requires longer. But it probably turns out a lot of these acquired arrhythmias also have a genetic component to it, increased predisposition to a certain drug, inducing the arrhythmia. The other part of the question about ischemia reperfusion injury, that's a little trickier, and it's not a place where we're likely to go to create ischemia within a tissue chip. It's really complex, it's really hard to control, but it's a good question.
Right. Robert Tsushima says, great talk, Glenn. How does the troponin I mutation prevent electrical capture in the iPS cardiomyocytes at the higher stimulation frequencies?
It's a great question.
Yeah.
And I was going to ask him the same thing. No. So we are working with James Weiss and Jilin Chu at UCLA, and they have a variety of models. We think one part of it is the fact that the off-rate constant is reduced or it's affected and it's affecting calcium metabolism and or calcium fluxes in a way that's arrhythmogenic. So Jim Weiss has a variety of models of alternans that's produced by a mishandling of calcium. And so they've been using our data and we've been interacting with them, and we're still working through the process to answer Robert's question. How does mishandling of calcium result in alternans? And there's a lot of work on this, but we're trying to find out the specific reason with our variant why it's happening. And we're not there yet, but I hope we will be.
When I looked at those data myself, I just thought about the refractory period, and it seemed like, is there a possibility that the refractory period has become lengthened, and that would implicate potassium channels or something like that, or is that?
Well, I showed the restitution slide. So we didn't measure the refractory period, but we measured restitution, or the duration of the action potential. And what the R37C showed is that it didn't get shorter as we increased the stimulation rate. And that, in fact, may be related. But again, working with Jim's group, maybe we can get some answers to that. I'm not sure yet.
Okay. Back to Peter Bax on maturation of the cells. He's asking, have you tried the Heron et al. Method from Jalife's group to promote maturation, apparently?
We have not. And there was another paper that came out very recently from Christine Mummery's group in Leiden in Netherlands. And she is creating tissue organoids in which there's iPSC cardiomyocytes representing 70% of the cells, 50% are fibroblasts, 50% are endothelial cells, and they're showing clear signs of maturation that are due to the presence of these other cells and whatever cytokines they are releasing into the environment. And it's pretty impressive, but there's so many aspects to maturation. I haven't tried it. That's all I can say.
Yeah, okay, fair enough. So let me ask you a mitochondrial related question. When you're growing these cells in a dish, and are they, first of all, are they quiescent until you stimulate them? When you showed us that those cells were beating in unison there, it sounds to me like they have an automaticity. Do you have to stimulate them to get them to contract, or do they beat on their own?
Well, that's a good question. So typically these cells, these tissues actually, because they're a monolayer of cells, they typically beat at about 50 beats per minute. They kind of beat like a human heart. It's a bit eerie. But the fact that they're ventricular cells and they're beating just reflects their immaturity. And the inward rectifier is not expressed at a high level. Therefore, the cells are sort of depolarized, and they probably express more of the pacemaker channel than what you'd expect in a ventricular cell. So they do spontaneously beat. One of the aspects of these maturation protocols is the beating frequency goes down, and ultimately it should go to 0, and they should only contract when you stimulate them if in fact they represent ventricular cells.
I see.
But
okay.
When you make them with the Kyer protocol here, they spontaneously beat. And that's one of the first characteristics you see. After 12 days, the whole dish is beating.
Okay. Have you ever thought of actually stimulating them for a period of time and seeing if maybe that's not in your not of interest, but for me, we stimulate muscle cells and hope to induce more mitochondria in those muscle cells and study that process. But I imagine these cells already have a high sort of energy supply from mitochondria. They have a good complement of mitochondria when you grow them.
So one of the aspects of maturation is if you use like a seahorse assay or something to evaluate mitochondrial function, you can clearly see increased metabolic activity like free fatty acid oxidation, other things attributed to mitochondria, which increase with the maturation protocols. So the cells, as you develop them and start beating, have a relative paucity of mitochondria compared to a native heart cell, but that biogenesis occurs during the maturation protocol. And one way you can push it is by pushing free fatty acids on it. And it tends to help the mitochondrial proliferation for reasons that aren't entirely understood, but it theologically makes some sense.
Sure does, yeah. But one more question here from Dr. Shime. Have you ever thought to test multigenetic disposition, or would it be too difficult?
That's a great question. And there was a paper, I think in the last year, from the Gladstone Institute, and it came from a real life situation where they had healthy parents who gave rise to 2 children with a very serious cardiomyopathy. And it turns out that both children inherited a variant from each parent, and it would require both of those variants to be present. So the children were sick, whereas each parent had only one of the variants. And so it was a very unique and unfortunate situation where the children inherited different variants from each parent, and collectively that produced the cardiomyopathy. So that question that the person's asking is a really good question, and it's something that you can do with iPSCs. Do you see a phenotype with another genetic variant that might predispose or exacerbate the condition? It's not something we do now, but it's something we will be doing in the future.
Right, excellent. Any other questions from the audience, please, at this time? Doesn't look like it. Glenn, I want to thank you very much for being with us today. Maybe next time we can actually pay for the ticket and get you a hotel room in Toronto, and you can visit that high-level tissue engineering place and see if Peter and Robert are around and all that. Maybe we can actually work that out another time. What do you think?
I think, did you say something about a glass of Malbec or some sort of red wine? Did you mention that? I thought I heard it.
We can work that out too.
Okay, that would be great. That's worth the flight. I mean, I reduced my carbon footprint to 0 on this talk. I think next time I'll invest the carbon footprint into a glass of wine.
Yes, yes.